Thank you. Thanks, everybody. I am Courtney Klepac, the Coral Resilience postdoctoral researcher. I am speaking on behalf of Erin Muller, who hopefully is faring the storm well, and also all of our corals up in Sarasota and the Gene Bank, as well as all of our staff up there. So I am going to be attempting to synthesize all of the previous work before my arrival at Mote and looking into the complexity of ranking these resilient genotypes within a nursery restoration context. Since active reef restoration, we have gotten really good at growing out coral both on land and in water, and then also outplanting on the reefs. And this is thanks to the efforts of probably everyone here at this conference. And we've seen that within that first year where we have more than 95% survival with Acropora cervicornis specifically, and that's very promising news. But what's happening after that first year of survival? At smaller spatial scales with Acropora cervicornis, in this site particularly, we're getting great growth, fusion, and then a return to functioning or ecological functioning. But this isn't always the case. A recent study by Van Woesik et al. Modeled Acropora cervicornis all outplanting events along the Florida reef tract and found that after 1 year up to 2.5 years, probability of survival significantly decreases, and it's not restricted to just one site. This is across all restoration sites, all subregions of the Florida reef tract. In addition, Ware et al. Looked at the probability of survival as well, looking at a longer term, a longer outlook, and it looks pretty bleak. The probability of survival for Acropora cervicornis gets really low when we extend out into multiple years. When we look in that same little window that Van Woesik looked at, we have similar, for those in the back, we do have similar declines or similar probabilities of survival within that 1 to 2.5 years. So what's going on? Although we're putting out lots of corals, there's unabated global threats, such as increased frequency and severity of thermal anomalies. We've got decreased pH and ocean acidification conditions threatening the structure of reef habitats. And then we have the pervasive and persistent effects of disease, which is affecting all coral reef species within the wider Caribbean. Yet, despite all this, some corals are able to resist stress more than others. And from a restoration context, we want to take and harness these adaptive traits, these resistant traits, and we want to utilize that for thoughtful restoration practices. Within Mote's restoration cycle, and this is probably common to other restoration practitioners, the focus is primarily growing up, putting out corals, and harnessing and increasing genetic diversity. But how can we combine both adaptive traits, resistant traits, with increasing genetic diversity? It's complicated, it's tough. But this tiny little box within the restoration cycle is where resilience or resistance training can come into play. So we can look at these big, large global threats as well as focus on smaller local direct effects. At Mote Marine Laboratory, and this is before my time, for the past 7 years, they have been basically examining through experimentation genotype and phenotype responses of their nursery coral species using multiple genotypes within and testing the response within the facility. The traditional long-term exposure had been utilized to look at these 5 main coral nursery species, where 5 replicate ramets were exposed to each treatment. We did 4 factorial treatments of control, and we did ocean acidification or high PCO2, ocean warming, high temperature, and then the combined end-of-century predictions for ocean warming and acidification. 1 week of ramp up to temperature or ramp down in pH, followed by 8 weeks at that exposure treatment. At 0, 4, and 8 weeks, we took non-destructive measures of coral growth, measured various ways, as well as photophysiology measured various ways. At the end of the experiments, we did destructive measures of symbiont density, chlorophyll concentration, host protein, immune responses. So in these past 7 years, what have we learned? First, we've learned that stressors are often synergistic. So in this panel on the left, this is looking at growth for buoyant weight for Acropora cervicornis, 12 genotypes examined in 2016. We see a similar decrease in growth under the independent ocean acidification and ocean warming conditions, but a much larger concerted reduced growth under both of those stressors together. In addition, and similarly, bleaching susceptibility measured through total chlorophyll, we saw almost a negligible effect of OA. We saw a strong reduction or a strong bleaching response under temperatures, and then a much greater response when we look at combined stressors. Next, when we look at these traits for resistance or resistance traits, they often appear independent when you're looking at each one independently or in singular. In this heat plot of Orbicella fabulata exposed to end-of-century conditions, a resistant trait is going to be one that's colored in yellow, or if this trait is less resistant, it's going to be a darker color. And when we look at these 5 genotypes of Orbicella fabulata that are blocked out, you can see that it's not always consistent. If you are resistant or you have a more resistant score for your weight, you might not necessarily have the same resistant score for your photophysiology under stress. These corals were also exposed to stony coral tissue loss disease, and we see a similar disconnect when we look at disease resistance, where not the same genotypes are going to be disease resistant, grow well, or respond to heat stress with their photophysiology. So our goal is not to just find the responses, but look for winners and losers. And throughout this panel, it's difficult to tease apart the potential winners, yet we are able to find 3 potential, she did losers. Cute. We found 3 potential losers that have a concerted lower resistance score, lower resistant trait across the board. In addition to these traits, you can measure these traits in a variety of ways. So how you measure that phenotype, let's say the bleaching phenotype, can be measured through Fe over Fm, chlorophyll, RGB analysis. There's various ways. That can also differ depending on the experiment and the coral's response. So this was a study with 3 genotypes of Pseudodiploria clivosa. Let's focus on ocean acidification, where basically the ranking values for the 3 genotypes is going to be different when we look at Fe over Fm versus when we look at chlorophyll. Moreover, when we incorporate and look at high temperature or ocean warming, we find kind of like a winning and a losing genotype based on their Fe over Fm values, but that doesn't always translate into the same pattern or that same ranking when we're looking at chlorophyll. In addition, that type of experiment matters. So previously, all Mote has worked with has been these like long-term 8-week-long exposures. With my arrival, I brought in the acute coral bleaching automated stress system, or CBAS, because I was like, you guys have hundreds of genotypes. We don't have the time, we don't have the energy to stress test only 10 to 15 genotypes at a time. We can rapidly assess our nursery genets using a CBAS approach. And that's what the CBAS looks like. It's very transportable, and you can do various temperature stresses, 3-hour increase, 3-hour hold, 1-hour decrease, and then recovery overnight. And what we find when we compare our experiments with the response of Fe over Fm, we find that there is actually a strong overlap. About 70% of the 10 genotypes tested had similar Fe over Fm responses with respect to experiment. And we only found 3 genets that differed in their Fe over Fm response in respect to experiment type. Now, we've looked at various types of traits. We commonly are looking at now multiple stressors. So what are their potential trade-offs in all of these traits in our corals that we're experimenting on? So within the combined end-of-century context, this is basically an association plot where we have all of the traits measured within the combined ocean warming and acidification treatment. X's represent non-significant. Darker colors represent a positive association, and red represents a negative association. And what we can see is that in all of these traits that we measured, we found very few negative associations or potential trade-offs within this combined treatment. And we find more positively associations under our traits. Now, when we can kind of flip this on its head and look at each trait, we can look at all the treatments within each trait. We also find positive associations of the bleaching response, such as chlorophyll, when we compare all of our 4 treatments to one another. And then similar pattern when we look at, again, all of the traits, we do find that traits of genotypes do respond similarly under these different environmental treatments. Lastly, recent work by Klinges et al. She is kind of our geneticist, disease microbiologist. She's done some experiments where we have 2 genets of Acropora cervicornis that have a different microbial consortium that are disease resistant. So we were wondering, does this translate, if you're disease resistant, does this translate into potential loss of fecundity? Dr. Hannah Cook is in prep of putting this publication out, where when we look at those 2 disease-resistant genotypes, we see that there is no apparent trade-off in fecundity, especially when we look at the x-axis here as mean total eggs per bundle. And so these are the 2 resistant genotypes. They still have a high number of total eggs per bundle. Only one of them has kind of a lower sperm count per bundle. We have found some small instances of potential trade-offs, and I think this just depends on which species you're looking at. And it also depends on what you're measuring. So again, within the 3 genotypes of Pseudodiploria clivosa, we did find this inverse relationship between growth and bleaching susceptibility. But when we look at light calcification instead of growth rate, that potential trade-off is lost. But again, let's return back to the whole determining who's doing better or worse than others. And you can see that these red colors are a higher score, or higher z-score, and then the blue is a lower z-score. So again, we can actually kind of suss out when we are working with smaller genets, smaller number of genets, who the winner and who the loser might be. So everything that I've told you, it seems to be really complicated, right? And I think that collectively, all of this work that we've been doing with our genets of our 5 nursery species, all of our responses seem to be species specific, not really a huge surprise. So why bother doing all of this work that we've been doing? And why not bank on genetic diversity, which is kind of the original crux of restoration, was just let's enhance genetic diversity. So let's go back to that very first figure that I showed you. If we do not thoughtfully incorporate into integrating resiliency or resistant traits in our coral species, genotypes, or populations, then this is what we're going to see in the long term. Again, this is cervicornis. And these stressors, they're not going away. I mean, as we've seen this week, the stressors are only increasing frequency, severity. So we do need a plan for the future. So to summarize all of the work that has been happening, we do want to continue examining these threats in concert. These major threats are going to have synergistic or probably additive effects as well. And then resistance to one threat does not necessarily equate to a resistance for another threat. Those losers might be more readily easy to identify than the winners, which actually is probably good for maintaining genetic diversity, increasing genetic diversity, because we're not solely focused on let's keep that top performer, let's keep that top 10% of our genets that are doing the best. It's great to maintain genetic diversity, and so maybe we can just slowly figure out how to cull out the losers per se. As a community, I think, and Carly mentioned this yesterday, it's the biggest challenge is like finding these traits that are most informative, and then also making them consistent in their methodology and then in their reporting. But then I think also too, where especially a talk I was just at, I think it's good to look at multiple traits from a holistic approach and then see if those traits are heritable or plastic or adaptive. And I think that's the strongest point of moving forward working with these restoration corals. Trade-offs can exist, but the evidence is relatively limited, and I think it's probably specific to certain coral species. And lastly, I think just overall integrating these resistant traits in the context of active ecological restoration is going to be our best chance to restore resilient reefs in the long term. There are lots of people that have done this work, and there's lots of funding sources, and thank you very much.
We do have time for a question, if anyone has a burning question. I would like to ask you a question, and for anyone else gets the chance to put their hand up. So one of the key messages that I'm getting here, and I've seen it at other presentations as well, is obviously what you measure really carries huge implications into your decision making and kind of what it means.
Absolutely.
Where are we at in terms of informing, guiding what is being measured relative to outcomes and goals of different restoration programs? Is that something that is emerging? Is that something as a community which should start thinking about?
Sure. I mean, is there a workshop for that? I think that'd be
Let's talk.
I think that'd be really awesome, right? So I mean, there's so many things that we can measure. There's so many traits to measure, and then like when I think about bleaching susceptibility with just in that small, many faceted approach, it's like the responses I think are also related to the cell biology and the timing of a stress response. So I think it's still good to, and I think it depends on the practitioner itself. So like, not a plug, but we're giving a workshop next week, and we're going to be having participants that have low resources versus high resources. So I think it also just depends on your capacity to measure something. So sometimes simple as just survival or the color card, if you can make that a quantitative measure, then that would be really informative. But, you know, I don't know if there is a consensus yet. I know that everybody seems to have like their favorite flavors of things, but it's something worth looking into. And I think also not focusing just on the symbiont, but also the coral host, and then looking at the other participants in that holobiont as well.
I'd like to talk more about that even.
Much.
Great.
Cool.
Thank you very much. We'll move on to our next speaker. Thank you.